Good evening, friends, and welcome to ICAR JRF Plant Science online sessions. Thank you all for joining. Today, in this plant breeding session, we will be dealing about male sterility. Okay, so let's just start this topic. So, I hope that everyone knows about the definition of male sterility. Can anyone repeat it for me? What do you mean by male sterility?
Non-functional pollen.
Yeah, which means non-functional pollen. So male sterility directly or indirectly means that the pollen that is formed is unable to pollinate, fertilize, and ultimately it is not able to set the seed, right? So who discovered this male sterility? Do anyone have idea? Who discovered the male sterility? Male sterility was also discovered by the scientist who discovered your self-incompatibility, that is Goldritter. And we already said in modes of reproduction and pollination class that both self-incompatibility and male sterility, it promotes xenogamy or your cross pollination, right? And see, at the end of this session, you will be knowing how it can be used in breeding. Okay. So in today's class, we will be dealing about what are the different types of male sterility in croplands and what are the genes responsible for them and how you can transfer the male sterility, and it's important in hybrid production. Okay, so male sterility, it is based upon the phenotypic class, it is divided into three types. Number one is your structural male sterility. So the name itself, it clearly indicates that there is some defect in the structure of the male reproductive organ, so which means that the stamens on the plant, whether they are absent or they might be malformed or they are modified into different floral parts. Okay, so this comes under the structural male sterility. So structural male sterility means completely the stamens might be absent or the stamens might be formed but they are malformed, or we can say malfunctional, and the formed stamens are completely modified into the other floral parts. And the next is your sporogenous male sterility. Okay, so the name itself indicates that it is sporogenous, which means the defect is present at the sporogenous stages. So here the stamens are normal, the stamens are formed and the stamens are normal. But the thing that is abnormal is your microsporogenesis, the process of formation of your microspore, or either the pollen mother cell itself is your abnormal condition or the microspome that formed might be in the abnormal condition, right? And then coming to the other type is your functional male sterility. So everything is normal in case of your functional, which means the stamen is formed, your pollen mother cell as well as microsporogenesis, everything is fine. But the ultimate product, that is your pollen, okay, which is non-functional in nature, right? This is known as your functional male sterility. So based upon the phenotypic classes, male sterility is divided into three types: structural, sporogenous, and functional. Okay, so in case of structural male sterility, the structure of your male reproductive organ is either absent or malformed, or it has been modified into any other part. Whereas in case of your sporogenous, every the stamens are normal, but the microsporogenesis or your pollen mother cell or the microspore is abnormal in condition. Whereas in case of your functional, the stamen is formed, the stamens are normal, as well as your microsporogenesis, everything is normal. But ultimate product, what is your pollen? It is non-functional in nature. Okay. So please to remember, this is also a different class of your male sterility, that is your phenotypic class of male sterility. And can anyone say what are the three classical classes of your male sterility? How do we distinguish them?
GMS, CMS and CGMS.
Okay, so what do you mean by GMS?
Genetic male sterility.
Okay, see, these other different types of your male sterility, these are classified based upon the gene location. Okay, where the gene is located, where your gene that is causing your male sterility is located. Okay, so first is your genetic male sterility, cytoplasmic male sterility, cytoplasmic genetic male sterility, and transgenic male sterility. So whereas in case of your genetic male sterility, the name itself indicates our gene is located in nucleus, right? Whereas in your cytoplasmic male sterility, the genes are located in your cytoplasm. In case of your CGMS, that is the third type, both it is located in your cytoplasm as well as your nucleus. And the last one, transgenic male sterility, which means you will be using the genes from other organisms, okay, to cause male sterility in the croplands. Coming towards your genetic male sterility. See, male sterility, as I told you, these are governed by some genes which are located in different parts of the cell. So based upon its location, if it is located in case of your nucleus, so we call it as genetic male sterility. Genetic male sterility also has another name. Can anyone say it? Which is also known as your genic male sterility. Okay, both genetic as well as genic male sterility, both are your same terms. So genetic male sterility, it is actually governed by a single recessive gene. Okay, you need to remember this. It is governed by a single recessive gene, small ms. Okay, most of the male sterilities are governed by the recessive genes, but we have one exceptional case where you will see the dominant gene causes genetic male sterility in case of safflower. Okay, so we also discussed that this safflower is an example of your often cross pollinated crop, which is your oil crop. Okay, so please to remember, most of the times male sterility, genetic male sterility is governed by a single recessive gene, that is your small ms. But an exceptional case where genetic male sterility is governed by your dominant gene, that is your capital ms, is seen in safflower. So now we will see the inheritance pattern of the genetic male sterility. As I told you, it is governed by a single recessive gene. So always we will write female on the first side, right? So this is female and this is male. So male sterile plants, they generally have recessive genes in homozygous condition, and when these are crossed with your male, that is dominant homozygous condition, the F1, it will be in heterozygous condition. Okay, so whether this F1 will be male sterile or male fertile, how the F1 will be?
Okay.
Okay, fertile. So because it is fertile, because the dominant gene, it causes your
Dominant.
fertility. Yeah, and the recessive gene is being suppressed by this dominant gene, so it will be fertile. So when you self the F1, you will get three different genotypes. So can I know the ratio, genotypic ratio of these three different genotypes? So it will be in this condition, right? 1:2:1. So among these three different genotypes, which is the male fertile one and which is the male sterile one?
Last
Homozygous
one is
recessive
male.
is correct.
Okay, so the
Last
last
one is
one is
male
your,
sterile.
okay, last one is your male sterile one, right? And the remaining will be your male fertile, right?
Yes,
So
sir.
if we compare the ratio between male fertile and male sterile, it will be 3:1, right?
Yes, sir.
So in case of your GMS, when you cross a homozygous recessive into homozygous dominant, you will be having F1 which is in heterozygous condition, and when you self them, you'll be getting 3:1 ratio, which is similar to that of your mono hybrid ratio of your Mendel cross. Okay, so among these, 3 will be male fertile and the other will be male sterile, right? So did you get this point?
Okay.
Okay. So this is the inheritance pattern of your genetic male sterility. See, do remember that these small ms as well as your capital MS, these genes are located in nucleus. Okay, this is also an important question in your JRF. These genes, these are located in the nucleus. And now coming to how do you maintain the male sterile line then? See, if you cross a female with your male, so we can call this female as male sterile, right? And this as your male fertile
Yes.
line. So if you cross this male sterile with your male fertile line, always you will be getting male fertile.
Okay.
So how do you maintain the male sterile line? So we will see how to maintain a male sterile line. So when you cross this homozygous recessive male sterile line with your male fertile line, which is also a homozygous dominant one. Okay, so this is your homozygous recessive and this is your homozygous dominant. You will be having an heterozygous progeny, right, which will be male fertile. Okay, so when you cross this heterozygous male fertile progeny with the female parent again, so what do you call this cross?
Background.
Yeah, you will be performing a back cross. So when you perform a back cross, you will be having two different type of progeny. Okay, so one will be again male fertile and another will be the male sterile. Male fertile and male sterile will be in a ratio of 1:1. Am I right? In case of your back cross,
Yes,
when you
sir.
back cross the heterozygous male fertile with your female parent, okay, so you will be getting the male sterile and male fertile in 1:1 ratio. Okay, so you need to
Yes, sir.
identify the male sterile one, right? You need to identify the male sterile one. If you want to use the male sterile plant in your hybrid breeding program, you need to identify it. Okay, so this is the simple maintenance program. Again, if you want to obtain the male sterile one, what will you do? You can cross the male sterile one with this plant,
Male
right?
fertile.
Yeah, so male heterozygous condition. See, as these are being the progeny of a single cross, can we call them as SIBs? Am I right? We can call them as SIBs, right? Because these two belong to the progeny which are obtained from a single cross. Okay, so we can call them as SIBs. So if you want to maintain the male sterile line in genetic male sterility, you need to perform SIB mating. See, everyone are clear with this? We can call them as SIBs, right? So if you mate between them, that is, if you cross the male fertile one with the male sterile one, which are SIBs, so again you will be getting similar to 1:1 ratio of male fertile and male sterile. So again you will be crossing between these two and you will be maintaining the male sterile line, right? Is it okay?
Yes, sir. So how can we identify that? Like,
Yeah, see, male sterile line you can identify based on these, what do you call, the statements will be some type of a malformed, or they will be modified into in most of the cases they will be modified into some other floral parts. Okay,
okay.
okay, or these all male sterile lines, they can be identified before flowering itself. Okay, so you can easily
Okay.
identify based on them if you want to use them in case of your hybrid breeding program or in hybrid seed production. Okay, based upon these
Okay.
structures you can identify them and then you can proceed for the further process. So this is your genetic male sterility. One of the most important question in genetic male sterility is male sterility, which is governed by the dominant allele, can be seen in safflower. Okay, so please to remember this one. And the maintenance of your male sterile line is done by sib mating technique, right? And this is the entire case of your genetic male sterility. And now we will see what are the different types of your genetic male sterility. So in case of your genetic male sterility, there are classically two types. One is temperature sensitive genetic male sterility and photoperiod sensitive genetic male sterility. So we can simply call them as TGMS and PGMS. Okay. See, this TGMS and PGMS, these are mostly seen in rice. Okay. So whereas in case of your temperature sensitive genetic male sterility, there is a threshold temperature which is 23.5 degrees centigrade. So if a plant exposes above 23.5 degrees centigrade, so what will be the fertility status of the plant? How it will be? If the plant is above this threshold temperature, okay, above 23.5 degrees centigrade, then the plant becomes male sterile. Okay, and if the temperature is below 23.5 degrees centigrade, then the same plant will be male fertile. See here, same plant, the plant is same, but everything depends upon the temperature, and hence the name temperature sensitive genetic male sterility. Okay, so if this plant is exposed to a temperature of 24.6 degrees centigrade, then it will be male fertile. If it is at 23 degrees centigrade, then it will be, okay, sorry, if it is at 24.6 degrees centigrade, it will be male sterile. And if it is 23 degrees centigrade, then it will be male fertile. Okay. So in the similar way, there is another type which is known as your photoperiod sensitive genetic male sterility, which means everything depends upon the photoperiod. That is, which means your length of the day, right? Length or duration of day, day as well as your night. So the threshold photo period is 13 hours and 45 minutes. So if the plant, if it is above this 13 hour and 45 minutes, it will be male sterile. And if it is below this 13 hour and 45 minute of your photoperiodism, then it will be your male fertile. The same plant, please do remember this, it is the same plant. Okay. So two line system, I'll say this at last, okay, why it is known as your two line system. But up to now, just remember that everything depends upon the temperature as well as your photo period. Okay, so the threshold temperatures or threshold temperature as well as your threshold photo period is also important. You need to remember this. So among the temperature sensitive genetic male sterility and photo period sensitive genetic male sterility, which one is most preferable? Do you have any idea about which one is most preferable? Anyone?
PGMS.
Which one?
PGMS.
You can elaborate it in the full form. Which one?
The temperature sensitive.
Yeah, temperature sensitive. See, temperature sensitive is more preferable when compared to photo period sensitive. Okay, because the variation in the length or duration of your day and night, it only varies with your, what do you call? It varies with your longitude. So which is not possible in India. Okay, but India, we also know that it has wide range of temperature. Okay, because temperature it generally varies with your latitudes. So most preferable one is your temperature sensitive genetic male sterility, right? So please to remember, in India, temperature sensitive genetic male sterility is most preferable. Am I right, guys?
Yes, sir.
No. See, photoperiodism, it changes with your latitude, okay? Okay, photoperiodism, it changes with the latitude. But your temperature, it depends upon the longitude. So please to remember this. So this is the reason why our temperature sensitive genetic male sterility is most preferable in India. So hope you now got entire idea about genetic male sterility and then its types. So is this okay? Hello?
Yes, sir.
Can we proceed to the next type of male sterility? Okay. Let's see. And cytoplasmic male sterility. So whereas in case of cytoplasmic male sterility, the name itself indicates that the genes are located in your cytoplasm. So cytoplasm, and can anyone say that when we cross a female with male in the progeny, where does the cytoplasm come from? Whether it comes from the female parent or from the male parent.
Female parent.
Yeah. So every time, whenever you cross a female parent and male parent in the progeny, the cytoplasm is always from the female parent only. So please to remember this. Female parent contributes cytoplasm, and this cytoplasmic male sterility is mainly due to the mitochondrial genome. Okay, which means your mitochondrial DNA is mostly responsible for the male sterility. As we know that mitochondria is always present in the cytoplasm, as it is always present in the cytoplasm. So mitochondria will be eventually carried out to the progeny when you cross a female with the male, and then the progeny will also have the mitochondria from the female side. Okay, so this is the basis. Please to remember this. And when you see the inheritance pattern of cytoplasmic male sterility, so the name itself indicates that male sterility is caused by the genes that are present in the cytoplasm, and more particularly, it is due to the mitochondrial genome. So, when we have a male sterile cytoplasm, which is crossed with a male fertile cytoplasm, which means this acts as your female and this acts as your male. Okay, so in the progeny, cytoplasm will be always from the female side, right? But when we see the condition of the nucleus, so what will be the nucleus condition? So 100% in the progeny, 100% of the cytoplasm will be from the female side, and when we see the nucleus condition, 50% will be from the male as well as 50% will be from the female. Okay, so male and female contributes equally. So as the cytoplasm completely comes from the female side, so even the progeny, what will be the fertility status of the progeny, whether it will be male fertile or male sterile? how will be the fertility status of the your progeny when we cross a female with male, where the female is having the sterile cytoplasm? So the condition of your progeny will be
Male sterile.
male sterile, because
Cytoplasm is received from female.
yeah, 100% of the cytoplasm is received from your female, and female has the sterile cytoplasm, right? So the progeny will be 100% male sterile. Okay, and see, now we just saw about what will be the inheritance pattern of your cytoplasmic male sterility. You know, there are different lines to designate in male sterility. One is known as your A line, and the second is your B line. So what do you call this A line, whether it is your female line or male line? And what about the B line? How would you identify
So
them?
A is female line and B is male line.
Okay, A is your,
Female line.
so which is your male sterile, right? And then
Yes.
B is your male fertile line. Okay, right. B will be your male fertile line. So I have a see, these are two A line as well as your B line are two different strains, and there is also a particular term which is common between these two. Do anyone know A line and B line?
Isogenic line.
Yeah, what do you mean by then isogenic lines? Isogenic.
Iso means similar, genic means matlab gene constituents except one character.
Hmm. So nearly similar lines except for one or few genes. Okay, nearly all the genes will be similar except for one or few genes, whereas in case of our condition it is the male sterile gene. Okay, so this A line and then the B line are isogenic lines except for the male sterile line, which means your A line is male sterile but the B line will be male fertile, right? So if you want to transfer this cytoplasmic male sterility from one line to other line, okay, for example, let us consider you want to transfer the cytoplasmic male sterility from A to B only. You also want to make this B line also the male sterile one. Which method do you choose?
Backcross method, sir.
Yeah, so you need to perform continuous back crosses, right? See, the name itself indicates that cytoplasmic male sterility, which means the cytoplasm will be in sterile condition. So for example, assume that this is a female line which has the sterile cytoplasm, and this is the line that we want to convert it into the CMS line, which we want to convert. We want to convert. Okay. So what will you do? You will cross the male sterile, that is your A line, with your male fertile, the line that which you want to convert. So the progeny, how it will be? It will be male sterile, as 100% of the cytoplasm will be from the female side. And then you will again back cross this F1, that is the progeny that is obtained between the two parents, with which line?
Male fertile.
Yeah. Why you need to cross with the male fertile one?
Because we need that as a gene.
So we need only the gene from the male fertile, because see, cytoplasm will be always from the female side, but when we see the nuclear condition, it will be 50% from the male side and 50% from the female side, right? Male plus female. So whereas in case of your F1, it will be 50% from the male and 50% from the female. So when you again cross your F1 with the male fertile, that is your B line, that is the line which you need to convert into CMS line. So now what will happen? 75% of the nuclear status will be from this line, that is your B line, and 25% will be from the female line, and the cytoplasm will be always from the female parent. So if you go on back crossing this male sterile line with the line that you want to convert, after your 7 to 9 back crosses, you will be having a line which is very similar towards your B line. But what will be the fertility status of this B line? It will be male sterile, right? So if you go on back crossing it, so first it was 50%, next it will be 25 and 75%. And what will be the next condition then? 12.5 and 87.5. So ultimately your female nuclear status will reduce by half based on every cross, and then it will also increase by half. Okay, so with this method, with the help of your back cross, you can transfer the cytoplasm, that is your sterile cytoplasm, from one line to the other line, or whatever the plant you, whatever the line you wish to. Okay, so this is the method of your transfer
Sir,
of. Yes.
sorry, the increase and decrease is related to only nuclear only.
Yeah, only nuclear,
But
because
outside it,
no cytoplasm. Cytoplasm will be always from your which side?
but how it is related to the decrease and increase in nuclear status? How
See,
it's related?
cytoplasm will always come from the female side, but let me clarify. See, if you observe this picture, na, carefully see this picture. Okay, so the dotted one, this is your female parent. This is your sterile cytoplasm. Okay, and this is your male one, which is your fertile cytoplasm. But when you see the nuclear condition, for example, you just assume the nuclear condition of your female is X and the nuclear condition of your male is Y. Okay,
Okay.
when you cross your male sterile, that is your female, with your male fertile, male, it will be female. So when you cross between them, the cytoplasm always comes from your female side. Okay,
Okay.
in any organism, it will be always from the female side. But the nuclear condition, how it will be means, it will be half of your female side, that is your X, and half will be from the male side. Okay,
Okay.
so when you again cross this F1 with this male fertile line, which is known as your back cross, so what happens? The Y genome, it increases, right?
Okay.
So it again increases by half. Okay, so
Okay.
you just see here, first it will be 50 and 50%. So male, it will be your female, it will be your male, 50 and 50. Next your female, it will get reduced to half. So what is the half of your 50? It will be 25. Okay, and then in the next generation it again becomes half, and in the next generation it will again become half. So after your 6 to 7 back crosses, what happens is 99% will be your male genome. Okay, 99% will be from your male genome, but the cytoplasm will be from your female side, and that cytoplasm will be sterile. So which ultimately cause that it is your male sterile line, but this male sterile line is similar towards your male fertile line, okay, in all the genes except for sterile gene, okay. Is it okay?
Sir, cytoplasmic matrix is gene.
No, you need to be clear, okay. Tell me once again.
Sir, the male sterile gene will be located in mitochondria.
Yeah, where will be the
Mitochondria.
mitochondria present? Yes,
In cytoplasm.
mitochondria will, so cytoplasm. So which means always the mitochondria as well as cytoplasm will be coming from which side?
Female side.
So female side, okay.
Female side.
So what will be the nuclear condition then? If we see the nuclear condition, how it will be?
Nuclear condition is male
50%
cell. Male
nuclear
nuclear
condition will
condition is
be
male side is dominant, sir. After back crossing.
after 6 to 7 back crossing, it will be 99.99% from your male side, okay.
Okay.
Yeah, you need to remember this.
But
After, yeah,
how will, but how it is male sterile is introduced into male introduced in male fertility
yeah,
with the
this
change of
is this is
nuclear gene.
yeah, this is what now we will call it as your transfer of cytoplasmic male sterility. See, uh, if you read hybrid seed production, so for hybrid seed production, na, you need to have two lines. One will be your female and male.
Okay.
So hybrid seed is nothing but your F1 seed, okay? F1 seed. So if you want to have a successful cross between these two, what you need to have? If the female is having bisexual flower, which means both the male and female floral parts are in the same flower, okay? So can
Okay.
you ensure 100% cross pollination every time?
No, sir.
No. So this is the reason why we are using the male sterility. So if this bisexual flower has male sterile stamen, so there will be 100% cross pollination, right? There is no chance of self pollination then.
Okay.
Okay, this you
Okay.
understood. Okay, for
Okay.
example, yeah, another reason is why we need to transfer the CMS. See, we have developed a line which has all the essential features so that it can be the female line in your hybrid seed production. Okay,
Okay.
we have once developed a line which is suitable for female plant or female parent, okay, but the only condition is it, it has a flower where stamen as well as your pistil are side by side. So can you use this plant as your female in hybrid seed production?
No, sir.
Why can't you use it?
Sir, it's a self pollination.
Yeah, there
Self
is a chance
pollination.
of self pollination and your hybrid seed gets, what
Contamination.
will it get? Contaminated, right? So
Okay.
okay, so now we need to introduce the male sterile system into this line, okay.
Okay.
So you have one source, so just assume that this is your source, and now you need to convert this, you need to transfer this sterile cytoplasm into the plant that you wish to use as your female parent in hybrid seed production, okay.
Okay.
So this is the reason why we will be transferring the male sterility. Am I clear?
Okay. Okay, sir.
So this is okay. Is this your doubt?
Yes, sir, it is CUET.
Okay. So this is your cytoplasmic male sterility, right? So in case of your cytoplasmic male sterility, the F1 will be always male sterile, okay? And the reason being, cytoplasm will always come from the female side, okay? There will be an assertion reason type of questions in case of this. So assertion will be in cytoplasmic male sterility, F1 will be always male sterile, and reason is the cytoplasm will always come from female side, okay? So then what will be the answer to this? Are the both statements true or wrong? What will be the answer? Assertion is F1 will be always male sterile in case of your cytoplasmic male sterility, and reason is the cytoplasm, it always comes from the female side in your cytoplasmic male sterility. So both the statements are true, right? And the reason, it correctly explains the assertion, okay? So this type of questions might come in case of this CMS system. And then we'll be dealing with cytoplasmic genetic male sterility. See, just from the name itself, you can decide that the genes are present in both your cytoplasm as well as your nucleus, okay? Cytoplasm as well as nucleus. This is your CGMS system. CGMS system is almost similar to the earlier discussed cytoplasmic male sterile system. The only thing is that the restorer gene, okay, the restorer gene is available. There was a question many times in JRF, where is this restorer gene present? Do anyone know where is the restorer gene present? Whether it is present in your cytoplasm or in the nucleus? Where it is present, the
Nuclear.
restorer gene? Yeah. So please do remember that the restorer gene is present in the nucleus. Okay, this is the condition of your restorer gene. Capital R, capital R. Okay, restorer gene is present in the nucleus. CGMS system is similar to your CMS system, that is your cytoplasmic male sterility. The only thing is that your restorer gene is present in the nucleus. And see, the restorer gene, it will be in the dominant homozygous condition. Okay, the restorer gene is dominant, and hence we represent it by the capital letter. Okay, so now we will see how this restorer gene is being used in case of hybrid seed production, and how it can be transferred to different lines. See, we have one line where the cytoplasm is sterile as well as the restorer gene is in the recessive condition. Then what will be the status of this line? It will be always in male sterile condition, right? Because the cytoplasm is sterile, and then the restorer gene condition is also in the homozygous recessive form. So this will be male sterile. So when we cross a male sterile which has recessive restorer gene condition with the male fertile which has the fertile cytoplasm, but again the restorer condition is in the recessive condition, the ultimate progeny, it will be male sterile itself, because the cytoplasm will be coming from the female side, and one R allele is from the male side and the other R allele will be from the female side. So again the nucleus, the restorer gene position will be smaller and smaller, which is your homozygous recessive. Okay, and then this is the sterile cytoplasm. So the progeny will be 100% male sterile, right? Is it clear?
Yes, sir.
Okay, and then you can see another cross below. So the similar condition, the male sterile line is fixed in all the three crosses. Okay, the male sterile line. So when you cross this male sterile line with a male fertile line, But now in the nucleus, the restorer gene condition is homozygous dominant. Okay, and the cytoplasm condition is it might be either your fertile or it might be either your sterile. Please hear carefully. If the restorer gene condition in the nucleus is homozygous dominant, okay, and if the cytoplasm, it might be in either case, it might be in female, it might be in fertile, or it might be sterile. Ultimately the plant will be male fertile. Okay, did you understand this point?
Yes, sir.
Okay, so when you cross a male sterile line with the male fertile line which has the dominant homozygous dominant restorer gene, so where will you get the cytoplasm from? The cytoplasm will be from
Female.
female side. Then what about your nuclear condition? How it will be? 50% will be from your female side and 50% will be
Female.
from your male side. So that is the reason why we got dominant allele as well as your recessive allele. Okay, so as we know that dominant, it will suppress your recessive. Okay, and then it expresses. So the restorer gene expresses, which states that this restorer gene, it suppresses your recessive allele and it expresses. And also the restorer gene, it also suppresses the sterile cytoplasm. Okay, it has also the capability to suppress the sterile cytoplasm, and ultimately the progeny will be 100% what it will be,
Fertile.
100% male fertile. Okay, male fertile. Whereas in case of this cross, the progeny will be 100% male sterile. This cross is directly, or what can you say, this cross is your classical example of, this cross is the classical example of your CMS, right? This is completely your
Yes, sir.
CMS without any change. Am I right?
Yes, sir.
This cross. And when you come to this cross, what happened? You have got the restorer gene which is present in the nucleus in the homozygous dominant condition. And just leave about the cytoplasm, whether it is fertile or sterile. If you have a dominant restorer gene, then your entire plant will be male fertile. And when you cross your male sterile line with your male fertile line, the progeny, which even though it has the sterile cytoplasm, but it has a single R allele, even a single R allele is enough to suppress your recessive one and also to suppress the sterile cytoplasm, and then the progeny will be 100% male fertile. Okay.
Yes, sir.
Okay, and now we will see a third type of cross where the male sterile line is again fixed, but now the male fertile line, let us assume whatever might be the cytoplasm, it might be your fertile or it might be your sterile, but the nuclear condition is what we can call it as your heterozygous, right? Heterozygous
Yes.
restorer gene condition. So when you cross your male sterile line with your male fertile line, then the cytoplasm will be always from the female side. Okay, so from this you will be getting R gamete, and from you will be getting two different type of gametes. One will be your R and the other will be your small r. So when this R fuses with capital R, so it will form a heterozygous condition again. And when small r fuses with small r, it will be your homozygous condition. Okay. So among these two, which will be your male sterile one?
Homozygous. Yes,
Yeah,
yes.
the homozygous one. Because there is no dominant small capital R allele, right? It is absent. So as a result of this, and even the cytoplasm is sterile, so this will be your male sterile progeny. But in case of this, there is a single R allele. This R allele, it can suppress your recessive and it can also suppress the sterile cytoplasm, and it will be eventually male fertile. Okay. Is it right?
Yes, sir.
So whereas in this type of cross, the male fertile and male sterile ratio will be 1:1. Okay. So whereas in case of this, it is 100% male fertility, no 100% male sterility. Whereas in case of this, it is 100% male fertility. But in case of this, we can say 50% male fertile and 50% male sterile. Okay. So cytoplasmic genetic male sterility is a system where you can get all the three types. One is your 100% male sterility, next is your 100% male fertility, and third is your 50% male fertility and 50% male sterility. Okay, is it clear?
Yes, sir. Yes.
Okay, and now we will see how to transfer this restorer gene, okay, into a new line. So what is a method that we use to transfer a gene from one plant to another plant or from one strain to one other strain? What do you call that method? Backcross, right?
That-
Yeah, backcross breeding is a method where we can use to transfer the genes. Gene transfer always involves your backcross breeding program. Okay. So there are two strains. One is your strain A and next is your strain B. In case of your strain B, there is the restorer gene. In strain A, there is no restorer gene. So if you want to transfer this restorer gene from strain B to strain A, the method that you use is your backcross. So when you cross between these two, what will be the condition of the F1, whether it will be male sterile or male fertile?
Fertile.
In case of your CGMS, it will be fertile. Why? Because there is a single dominant allele.
Dominant.
Okay. Yeah. And where do you want to transfer this restorer gene? Into strain A, right?
Yes, sir.
So if you want to transfer this into strain A, what you need to do is you need to continuously backcross with your strain A. Okay. You need to
Yes, sir.
continuously backcross with your strain A, right? After 6 to 7 backcrosses, what you will be having is this line. Okay. After 6 to 7 backcrosses, you will be continuously having the, see, for example, there is RR genome into capital R, capital R. It will be heterozygous condition. Okay. And then again, if you backcross it with your small r, and again capital R, small r, again with this, and then you will be getting this. So now tell me, guys, which genome increases, whether it is small or is it capital R? Which genome increases after 6 to 7 backcrosses?
Smaller.
Yeah, smaller genome eventually increases. Why? Because we are continuously using the smaller as the backcross parent. Okay. So if we continuously do it for 6 to 7 generations, what happens is the ultimately formed nucleus will have 99% of the genome from the RR. Okay. And only the gene that differs is your capital R. Right? Am I right?
Yes, sir.
Okay. So once after attaining this type of the progeny, what you need to do is you can just self it. So if you self this, what will be the possible three type of your progeny? One will be capital R, one will be
Smaller, smaller. Capital
small
R, smaller.
r, and then the heterozygous condition, right?
Yes, sir.
So if you see, this is the same thing that they have got. and then among this, what will be the condition which is having smaller with the sterile cytoplasm? What will be the fertility status of this? It will be
Sterile.
male sterile, so it
Sterile.
can't be used. Whether the other two, they will be male fertile, right?
Yes, sir.
So male fertile. Among these two, there is one type where we can have homozygous dominant R alleles, right? This line is almost similar towards your strain A except for the gene capital R. Okay, is it okay?
Yes,
This
sir.
is 100% similar to strain A except for which gene?
Capital R.
What do you call that gene capital R? Restorer gene, okay?
Restore. Restore also.
Yeah, except for the restorer gene, right?
Yes, sir.
Is it okay? How you will transfer the restorer gene from one strain to other strain, right?
Yes, sir.
Which method will you use?
Backcross method.
Backcross method. It is also the similar case in the cytoplasmic male sterility where we will be using the backcross method to transfer the sterile cytoplasm into a new line, right?
Yes, sir.
Yeah, right. Hope you clearly
Excuse me, sir.
understood the CGMS as well as CMS system. See, CMS system is completely similar. No, CGMS system is completely similar to CMS system, except with restorer gene. Okay, if the restorer gene is available, then it gets converted into your CGMS. Okay, so this restorer gene, it is present in
Nucleus.
nucleus, which is also known as the brain of the cell. Okay, so these are important points. So this is one important question, that restorer gene is present in the nucleus, right? If the restorer gene is in whatever it might be, if it is in homozygous dominant or your heterozygous condition, whatever might be the cytoplasm, whether it is fertile or whether it is sterile, so what will be the fertility status of this line?
Fertile. Sir.
It will be male fertile, and it is used as
Yes,
your male line, right? Okay, right. So these are the
Excuse me,
three different. Yes.
sir. Can you please explain how to transfer a restore gene?
Okay, storage. See,
Sir,
you have
where
two.
we are using.
Yes.
Hello.
Yes.
Sir, we are transferring restore gene from strain B to strain A, right, sir?
No, no, no. You can transfer from any strain to any strain. Okay. Not only
Yeah,
from
but here we are
strain
using
B.
recurrent parent as strain A.
Yeah, we are using recurrent parent as strain A, right?
Yeah, and strain A contains sterile cytoplasm.
Yeah, sterile cytoplasm. Okay.
And
Then
sir,
proceed.
we have
What is?
used. Hello.
Yeah, tell me. Tell me.
So then in subsequent clauses, we have written fertile cytoplasm, right? Which we have used
Where?
as a recurrent parent.
No, see, I have just told
Yes,
you
sir.
example. We can transfer the restorer gene from strain B to strain A. Okay.
Yes, sir.
Did you? But we can also transfer the restorer gene from strain B to any other strain. Any other strain.
Sir,
Okay.
here, sir,
No, no,
recurrent
no.
parent is strain A and it is.
No, I just told you example to consider strain A. Okay.
Okay.
Just for example. But you can also transfer it to any other strain. Okay. See, this is any other strain. You can call this as any other strain. This strain, it is having the restorer gene in which condition? Recessive condition. Okay.
Yes, sir.
Okay, but the cytoplasm should be fertile. If the cytoplasm is not fertile, what it happens? If the cytoplasm is sterile, what it happens?
We can't use it as a female one.
Yeah, we can't use it as the female one. Okay,
Sorry, male.
see, A, B, these two are your isogenic lines. Please to remember. Okay, which means if A line has the recessive genome and also the sterile cytoplasm, then the A line also has the restorer gene in the homozygous dominant condition, whether it might be your sterile or fertile. Ultimately, what will be the fertility status of this one?
It will be fertile.
Male fertile, right? Okay, so A line and B line will be always, what do you call these lines?
Estrogenic lines.
Isogenic lines. Okay, so if we have a sterile cytoplasm for this one, for strain A, then we can also have the same line which have the fertile cytoplasm. Okay, if you go deep into hybrid seed production, you can know how these lines can be used. Is it okay?
Yes, sir. But can you please explain this example once again?
Okay, see, if you want to transfer the restorer gene from strain B, strain B will be always, how it will be? Strain B will be always male
Male fertile. Fertile.
fertile. Okay, so strain B will be always male fertile, and if
Yes, sir.
the, so what does it means? That it has a dominant restorer gene. Okay, is it right?
Yes, sir.
So, okay, if you want to transfer this restorer gene into any other strain, so first you will cross strain A with strain B. You will be having the F1, that is your progeny. It will be in the heterozygous condition. Is it okay?
Yes, sir.
And then this F1, it will be crossed with the line where you need to transfer this restorer gene. Okay, is it okay?
Yes, sir.
Okay, so just think whatever might be the strain it will be. Even you can have a strain where this is the condition, and if this is a sterile one, the cytoplasm is sterile one. Hello.
Yes, sir.
Okay, so if the cytoplasm is sterile one, so can you cross with these two?
You can't cross it.
So what should be the cytoplasm of the line that we need to transfer the R line? It should be always fertile. Okay.
Yes, sir.
Okay, so this is the condition where you can transfer the restorer gene from strain B, or the restorer gene, whether it is available, when it is available in homozygous dominant condition, you can transfer from one strain to any other strain. Okay,
Yes, sir.
so this strain will be used as the recurrent parent because we'll be again and again using the same strain. And ultimately what happens, this strain C, in this example it is given as strain C, and when after 6 to 7 back crosses, what it happens is, this strain will be having 99.99% of your genome similar to strain G, except for single gene. That gene is known as, except for a single gene, and that gene is your restorer gene. So once your restorer gene is now in a condition, in the background of your 99.99% of the strain C genome, you can just self this strain, and you will be having 3 different types of plants. One will be male sterile, you need to reject it, and the other two will be male fertile. But we need to only select the male fertile line which is in the homozygous dominant condition with restorer gene. Okay. So now tell me, how will you identify between these two? One is your heterozygous and other is your homozygous dominant. So how will you identify between these two?
Sir, we'll go for progeny test.
Yeah, we no protein testing. You just self them again. Okay.
Yes, sir.
So if you just self them, the heterozygous one will segregate, but the homozygous one will not segregate, right?
Yes, sir.
So you can fix that. This is the line which is 100% the strain C, not 100% but 99.99% strain C, which has the restorer gene incorporated or transferred, right? Is it
Yes, sir.
okay now? Are you clear?
Yes, sir. Got it. Yeah, I
Sure.
got yes, sir.
Okay.
Yes, sir.
So with this, we will be completing the 3 different types of male sterility. And so, do anyone have idea about transgenic male sterility? What do you mean by transgenic male sterility? What is you? What is known as transgenic male sterility? It is simple.
Male sterility genes come from other organisms.
Male sterility gene. Okay, we are taking male sterile genes from the foreign organisms, right? Organisms, right? So, see, male sterile means transgenic male sterility is also a type of your genetic male sterility. Okay, because ultimately we will be integrating the trans genes into the nucleus of plant. Okay, so now can we call this as a type of your genetic male sterility? Is it right? Right, we can call it as the type of your genetic male sterility because the trans genes which we are taking from the other organisms, we will be integrating it into the nucleus of the plant. So this is also a type of your genetic male sterility. So now in the transgenic male sterility, we will be seeing a system named Barnes-Barnstar system. Barnstar system of male sterility. Okay. So whereas in case of this Barnes-Barnstar system of transgenic male sterility, do you know which is the organism involved in this TGMS?
Bacillus amyloliquefacius.
Bacillus amyloliquefacius. Okay. So what are the genes that we get from these Bacillus amyloliquefacius? What are the genes that we got from this bacteria? We have got the 2 genes, one gene named as your Barnes, other gene named as your Barst. Okay, so there is also one other organism involved in this Barnes-Barnstar system. The organism name is your Streptomyces hygroscopicus. Streptomyces hygroscopicus. From this bacteria, we took a gene named Bar. Okay, Barnes and Barstar are from Bacillus amyloliquefacius, whereas your Bar gene is from the organism named Streptococcus hygroscopicus. Okay, and these two we will be seeing in the next slide how this system is used and which chemical is needed in this system and where and when it is needed. Okay, so just see, scientists after isolating a gene named Barnes, they have incorporated it into the anther of the plant. Okay, plant. This Barnes, what it does is it destroys your tapetal cells. Okay, It ultimately destroys the tapetum cells. When Barnase is incorporated into the anther of your plant, it destroys the tapetal cells. When the tapetum cells are destroyed, ultimately formed pollen grains will be male sterile. Is it okay up to this? When Barnase gene is incorporated into the anthers, it will ultimately results to the destruction in the tapetum, and then these plants, they the pollen which is formed will be male sterile, because tapetum is the tissue which nourishes your pollen grains. Okay, so when the tapetum itself is destroyed, so pollen grains do not get nourishment and male sterility is seen. Is it okay? Hello?
Yes, sir.
Okay, so tapetum will be destroyed and then the pollen grains will be in male sterile condition. And then there is another gene named bar star. Okay, this is your Barnase, which is in green color, and there is another gene named bar star. So this bar star, it is used to counter this Barnase. Okay, so when you inject only Barnase, tapetum is destroyed. When you inject both Barnase as well as bar star, this bar star will counter this Barnase, and the tapetum will not be destroyed, and tapetum is good, so the pollen grains get nourishment, and ultimately the pollen will be male fertile. The pollen will be functional and it will be male fertile. Right? Is it okay?
Yes, sir.
Okay. So this is the basis of this system, which is your bar star Barnase system of male sterility. Okay. See, this is known as the Barnase gene. And we also discussed that there is another gene which is named as your bar. This bar gene is from which organism?
Streptomyces hygroscopicus.
The bar gene is from your Streptomyces hygroscopicus. See, our A line was developed in such a way that there is only hemizygous condition of our Barnase and bar gene. Hope you know about hemizygous condition, which means there is only a single allele, right? Which is only a single construct. This is known as hemizygous condition, right? You need to have two alleles. One is your capital R or capital R or small r small r. If you have only a single allele, then what do you call this condition? This is known as your hemizygous condition. Okay, so this construct is incorporated in the hemizygous condition. So when this Barnase gene is incorporated into the anther, what will happen ultimately? The tapetum will get destroyed and then the pollen grains will be in male sterile condition. Okay, is it okay? Is it clear, right?
Sir, repeat.
Okay, see when you incorporate this Barnase gene into the anther, this Barnase gene, it is incorporated in the hemizygous condition into the anther. What happens? Barnase will result in the destruction of your tapetum. If tapetum is destroyed, there is no nourishment to pollen, no nourishment to the pollen. So pollen is non-functional, which means there is your male sterile condition. So now you got, if we introduce this Barnase gene into the anther, okay, the tapetum is destroyed, and if tapetum is destroyed, our pollen will be in male sterile condition. Is it okay?
Yes, sir.
Okay. So this construct is in the hemizygous condition. which means there is only a single construct. There is no counter to it. Okay, is it okay? So
Yes, sir.
we can call this as your male sterile line, right? Because there is Barnes gene. So when you cross this male sterile line, that is your A line, with the normal one, which is not transgenic, which is not non-transgenic, your normal plant, which has the normal construct. There is no transgene here, right? Everything this is your transgene. Is it okay?
Yes, sir.
Wait, I'll show you clearly. See, this is your transgenic construct, right? It came from other organism. Barnes came
Yes,
from
sir.
one organism which is known as your Bacillus amyloliquefacius. Bar gene came from your Streptomyces hygroscopicus. But this is in the hemizygous condition, which means it is only in the single one. There is no counter to it. Whereas in case of your non-transgenic, which means there is no transgenic construct at all. So this is your normal one, right?
Yes, sir.
Am I right? So just assume a condition
Yes,
where
sir.
you will be crossing capital R small r with your small r, where capital R is the transgenic construct. Is this similar, right? This is your capital R, this is
Yes, sir.
small r, this will be R,
Yes,
R.
sir.
Similar condition, okay?
Yes, sir.
So if you cross the transgenic constructed plant with your non-transgenic plant, you will be having male sterile and male fertile in 1:1 ratio, okay? Because this will go with this will go with this, and this with this, and this with this. So what happens ultimately? You will get male sterile and male fertile in 1:1 condition. Is it okay?
Yes, sir.
So one is your male sterile, another is your male fertile. So how will you identify? In order to identify, we need to spray a chemical known as phosphoenithricin, okay? Right? What this chemical
Yes,
does is it kills the male fertile lines. Why it kills the male fertile line? Because see, when you cross out the A line with the non-transgenic line, you will be having one this type of construct, the other construct will be this, right? There is no transgene here.
yes, sir.
See, this bar is the gene which confers resistance to this chemical. So when we spray this chemical to male sterile line, as there is bar gene, this bar gene, it will offer resistance to this chemical. But there is no bar gene here, right? There is normal construct. This is non-transgenic again in the progeny. Is it okay? Hello? Did you understand? Okay, I'll still simplify it. See, this is the construct, transgenic construct, which is in the hemizygous condition. Whenever you cross it with the non-transgenic plant, right? So what will be the two different type of gametes which are obtained from here? One, it will be the construct with the transgenic, other will be the normal one. From this, there is only one type of gamete, that is your normal one. So in the progeny, what happens is you can have one this type of progeny and next is this type of progeny. Is it okay now? Is it okay, guys?
Yes, sir.
Yeah. Okay. So this is having the construct and this is not having anything. So this will be again non-transgenic in nature and this will be transgenic because it has the construct. So if you want to identify which one is male sterile and which one is male fertile, you need to spray a chemical, right? And this chemical, it is known as phosphophenothricin. When you spray this chemical, the plant which do not have any construct, that is your non-transgenic one here, which does not have any construct, it dies. Okay, this plant, it dies, right? This plant survives. The reason is it has a construct gene called bar, and this bar confers resistance to this chemical. Did you understand?
Yes, sir.
Okay.
Yes, sir.
So this is the reason why the male sterile plants, after spraying the chemical, they are alive, and then the plant which do not have any transgenic construct, this is dead. Is it okay? If you understand this,
Yes,
now
sir.
we will see how the hybrid seed is, how it is used in the hybrid seed production, right? Okay. So up to now you saw this. When we see the hybrid seed production, see the male sterile line, it has the Barnase gene. Okay, it has the Barnase gene in the hemizygous condition. Is it okay?
Yes, sir.
Whereas your male fertile line, it has everything similar to that of your male sterile line, except that it is in the homozygous dominant condition, which means see, it is in both the constructs. Am I right?
Yes, sir.
Homozygous dominant condition. Now the Barnase gene, it is been replaced by a gene named as your bar star. Bar star, right? Bar
Yes,
star.
sir.
So when you cross between these two, so this is your female parent and this is your male parent. So there are F1s developed. Okay, this is your hybrid seed, right? See why the Barnase function here, it is been inhibited because bar star counterattacks the Barnase, right? Did you remember here? Bar
Yes,
star,
sir.
it counterattacks the Barnase, and then there will be no tapetum destruction. Tapetum will be normal. As the tapetum is normal, the pollen will be functional. Am I right?
Yes, sir.
So same is the case here. When we cross the hemizygous female plant which has the Barnase construct with your homozygous dominant male plant which has the bar star constructs, so the F1, where all the F1 will be male fertile because bar star will counterattack the Barnase. Here there is normal construct, there is no transgenic in case of this. So ultimately all will be fertile, right? All the F1 will be fertile. Is it okay?
Yes, sir.
Okay, so this is your Barnase Barnstar system of male sterility. Here you need to remember what are the organisms involved and what is the chemical used. Okay, what is the chemical name? The chemical name is your
Post
phosphoinositide.
phosphonothricin.
Yeah, and see which is the gene which confers resistance to this phosphoinositide?
Bar.
Bar.
Barging.
Okay, remember it is not your Barnase or Barstar, it is your bar gene, and this bar gene, it comes from Streptococcus hygroscopicus. Okay, bar gene, right?
Yes, sir.
Right. So this is the transgenic male sterility example, Barnes Marstar male sterility, and this system is also a type of your genetic male sterility because ultimately the transgene construct is incorporated into the nucleus of your plant, right? And do you know where this was first used? It is used in your rapeseed or even in your tobacco. Okay. So the scientist name who discovered this Barnes Bansar system, do anyone know it? Barnes Banstar system was discovered by, there was a scientist, I think so, Marini or something. Okay, So he was the one who discovered this Barney's bar star system of transgenic male sterility. Is it clear?
Yes, sir. Definitely.
Okay, right. With this, we will be completing the male sterility part. Right, we have completed the modes of reproduction. We have completed the uh, self-incompatibility as well as male sterility in detail. Okay, so hope everyone understood all the three basic and essential concepts that we need, right, so that we can enter into the breeding methods, right?
Sir, sir,
Yes. Yeah.
sir, please, please explain, sir, male gametocytes, sir. Only important parts of that.
Male gametocytes, na, you need to by heart them from the BDC book, man. You need to remember what do you call, there is a table, okay, which gene is no, there is a table belonging to the male gametocytes. You need to by heart them and you need to remember them. Okay.
Yes,
Okay, you have a table where there were divided into three different types of your male gametocytes. The what do you call that, I think it was the first generation, second generation, and the current generations. Okay,
yes, sir.
you need to remember all the male gametocytes that are listed there, and you also need to remember um, they belong to which class. I'll just tell you one important bit that was given in examinations. There is a male gametocyte. Let me write. There are two male gametocytes. One is your mg1 and mg2. Okay, these belongs to arsenicals. Among these two, na, these are being generally used in rice crop. Okay, mg1 and mg2. Among these mg1 and mg2, mg2 is most extensively used in China. Extensively used. And this mg2, its common name is sodium methyl arsenate. Okay, and then the mg1 common name was zinc methyl arsenate. Zinc methyl arsenate. Mg1 and mg2 are the male gametocytes which are used in rice, and the most used is your mg2. And there is also another male gametocyte which is known as your clofenset. This is also known as genesis. Okay, this was found to be the most ideal male gametocyte. Ideal male gametocyte, and this clone fact it is been used in wheat, right? So just remember this one, the mg2 rice, mg1 and mg2. Mg2 was most extensively used in China, and this mg2 common name is your sodium methyl arsenate. Is it okay? So what do you call these male gametocytes? There is also another name to them. Male gametocytes are also known as your CHAs, which are chemical hybridizing agents. Okay, is it okay?
Yes, sir.
Okay. So with this, we will be completing the male sterility, the most four important types, basically three: genetic, cytoplasmic genetic, and transgenic. It comes under the genetic male sterility itself. Okay, is it okay?
Yes, sir.
Okay. So with this, I will thank you and conclude the session here. Okay. Thanks all for joining, and we will meet in the next class.